Hello students, my name is Rahul Gudhe and today we are going to learn one small topic from 12th standard and that is nothing but the structure of the DNA. So when we talk about the structure of the DNA, so let me tell you for the first time DNA, it was the basic structure of the DNA. It was basically given by Watson and Crick. For the first time, these were the two scientists who had given the how the DNA basically it looks, okay, and the structure of the DNA how it can be. So that is what their theory is. So basically, when we talk about the structure of DNA, DNA is called as deoxyribonucleic acid. Some people call it as deoxyribonucleic acid, but that's a very wrong term. It is basically called as deoxyribonucleic acid. Okay. So if we talk about components of DNA, in that case, DNA is basically made of three different components. The first component is called as sugar. In case of DNA, this is called as deoxyribose sugar. Okay. Second component is called as nitrogenous bases. And the third component is called as phosphate group. So basically these are the three basic components of the DNA from which the DNA it is actually made of. These are called as the basic components of the DNA. Okay. Now we'll gradually try to see about each of them. So when we talk about deoxyribose sugar, if you don't know that there are two different types of uh, sugars which are present in case of nucleic acids. One is called as ribose sugar, another is called as deoxyribose sugars. In order to understand what is called as a deoxyribose sugar, we'll try to draw both the diagrams of ribose as well as deoxyribose sugar. Then we'll move ahead towards the structure of DNA so that you will also realize what is the difference in RNA and DNA. RNA is called as ribonucleic acid. Okay. So this is what I am drawing, and this particular sugar it is actually called as ribose sugar. Okay, this is called as ribose sugar. We'll try to differentiate between them, but please try to keep your focus on this particular thing. And right now what we are going to draw is called as, this one is called as deoxyribose sugar. Okay, and this is called as deoxyribose sugar. Okay. Now please try to see the difference between them. This is carbon atom 1, this is 2, this is 3, this is 4, and this is carbon atom number 5. Likewise here also, this is carbon atom 1, carbon atom 2, carbon atom 3, carbon atom number 4, and this is carbon atom number 5. If you see here at carbon atom second, here there is presence of OH group, correct? But if you see here, there is only H which is present. That means here oxygen is present. In that case, when it is called as ribose sugar, then when this oxygen is absent, that's why this particular sugar it is called as deoxyribose sugar. Okay, this is the first component of the DNA. Do you get my point? Carbon atom first, second, third, fourth, and fifth. This is called as ribose sugar. If you say at the carbon atom number 2, there is presence of extra oxygen. That's why it is called as ribose sugar. But when we see in case of the deoxyribose sugar, there is no oxygen present at carbon atom number 2nd. Getting my point? This is the difference in ribose sugar and deoxyribose sugar. Deoxyribose sugar, because it is present in the nucleic acid, That's why that nucleic acid it is called as deoxyribonucleic acid. And when the ribose sugar it is present in nucleic acid, in that case that will be simply called as ribonucleic acid. Okay, so this is basically the first component of the DNA. Second component was the nitrogenous bases. Okay, what these nitrogenous bases are? These are nitrogenous base. These are those compounds which basically contain nitrogen. For the time being, we'll not go in much detail about their structures. But if we talk about these nitrogenous bases, in that case, these nitrogenous bases they fall under two different categories. The first category it is of purines, and in case of purines we are talking about DNA, we are not going to talk about RNA. So in case of nitrogenous bases which are present in DNA, they contain purines. They are adenine, and second is called as guanine. These are both called as adenine and guanine, both are called as purines. Second group is called as pyrimidines. Okay, purines, and second group is called as pyrimidines. Pyrimidines, they are of two different types. These are called as cytosine and thymine. Okay, so nitrogenous bases, they are of two different groups, but they basically are four different: adenine, guanine, cytosine, and thymine. Adenine, it can be represented as A. Guanine, it can be represented as G. Cytosine, it can be represented as C. And thymine, it can be represented as T. So right now from here onward, we'll be considering only A, G, C and T. We'll not take their complete names. Okay. This is called as the second component of the DNA. The third component of the DNA, it is called as the phosphate group. Phosphate group, basically it comes from an acid, and that is called as phosphoric acid. If we talk about the phosphoric acid, how it looks, so basically phosphorus, it is attached on three different sides by OH, and on the other side it is having two different bonds with oxygen. You can write this phosphoric acid as H3PO4. This is simply called as phosphoric acid. This phosphoric acid is basically the third component of the DNA. Okay, so by far we have seen all the three components. Let's see how the structure of this DNA is. When we talk about the structure, two terminologies are there, or maybe you can call it as structural combinations in case of the DNA. So when we talk about the structural combinations, there are two different structural combinations. The first combination, it is called as nucleoside, and second one is called as nucleotide. What is a nucleoside? Very simple. We have three basic components of the DNA. So if you say that the first component, that is called as sugar, plus second component, that is called as nitrogenous base, when both of them they combine, in that case they form nucleoside. Getting my point? Nucleoside means sugar plus nitrogen base, first component plus second component. Okay, but in case of nucleotide, when with this sugar and nitrogenous base, if the third component combines, and that is called as phosphate group. Now in this case, it is called as nucleotide. In other words, you can just call it like nucleoside plus phosphate group. Okay, that is simply called as nucleotide. So these are the two different terms which we'll be studying ahead. Okay, so with these things, like what are the basic components of the DNA, and second, how they basically combine, let's see how the structure of this particular DNA is. Okay, now if we say, for example, ribose sugar, deoxyribose sugar, it can be represented like this. Okay, it can be represented like this. For your kind information, let me tell you, this is at the top of this, there is oxygen. This is considered as carbon atom number 1. This is 2, 3, 4, and this one is considered to be carbon atom number 5. Okay, this is simply called as deoxyribose sugar. Okay, nitrogenous base, sugar plus nitrogenous base is called as nucleoside. So how this nucleoside is formed? With the help of this deoxyribose sugar and nitrogenous base. Where will this nitrogenous base be present on this particular sugar? So it will always present at carbon atom number 1. Let's take for an example. This is adenine, which is present at carbon atom number 1. This is completely called as nucleoside. Second, maybe we can say like there is guanine, which is present again at the another sugar. This is one nucleoside, this is second nucleoside. On the third side, on the third part, there is presence of cytosine at carbon atom number 1 in this another nucleoside, and lastly again there is presence of another nucleoside. Okay, so these are the 4 nucleosides. Nucleosides along with phosphate, they are called as nucleotides. Correct. So let's see where this phosphate group is present. Phosphate group, it remains present between carbon atom number 3 of first nucleoside, or maybe first sugar, and with the fifth carbon atom of the second nucleoside or the second sugar. Likewise, another phosphate group, it will remain present here. Another phosphate group, it will remain present here. Okay. Now, this particular thing is called as nucleotide. This particular thing is again called as nucleotide. This one is also called as nucleotide. Okay. This is one strand of the DNA. But if you know that DNA, it is having double strand. Correct. Now, how this second strand will work? So please remember, there is one thumb rule so far as DNA is concerned, and this thumb rule says that A can form 2 hydrogen bonds with T, and G, it can form 3 hydrogen bonds with C. A, it can form 2 hydrogen bonds with T, and C or G, it can form 3 hydrogen bonds with C. Now, if this is true, in that case, please tell me, A, of course, it will be forming 2 hydrogen bonds with T, correct? Likewise, this G, it will also produce 3 hydrogen bonds with C. This C, it can form 3 hydrogen bonds with G, and likewise, this T, it can form again 2 hydrogen bonds with A. Correct? Now, before some time when we were talking, at that time I told you that nitrogenous base, it is always present at carbon atom number 1 of the sugar. That means this nitrogenous base, it must also be present at the carbon atom number 1 of the another sugar. That means carbon atom number 1, 2, 3, 4 and 5. Okay. Likewise, this C, it must also be remain present at carbon atom number 1. Likewise, this G, it must also be present at carbon atom number 1. Likewise, this A, it will also remain present at carbon atom number 1. Now, as here I told you that Phosphate group, it remains present between two nucleotides, but on one hand it remains attached with the carbon atom number 3, on the another hand it remains attached with the carbon atom number 5. If this holds true, same will be true here. This phosphate group, it will remain present between carbon atom number 5 and carbon atom number 3 of two different sugars. Same thing will even happen here. Same thing, it will even happen over here. This is how this particular structure it is formed. You can represent this particular structure like here. This whole structure, it can be represented like this also. Generally, you might have seen this in your books, but this is basically, it is done. Now, which carbon atom is here? It is called as carbon atom number 5. Which carbon atom is at this end? It is 1, 2 and 3. That's why it is the third carbon atom which is present. Likewise here, first, second and third. Here, first, second, third, fourth and fifth carbon atom is here. Okay. So the carbon atom which is present at one end of this particular DNA strand, this is 5, and that's why on the another hand there is the presence of 3. That's why this direction of the DNA, it is considered to be 5 dash to 3 dash. It is also said as 5 prime to 3 prime. But if you see on the another hand, this 5 prime to 3 prime direction is something different. One is going in this direction, another is going in this particular direction. Correct? That's why DNA is said to be polynucleotide double stranded, where strands they are anti-parallel to each other. These are one of the specialties of the DNA. That's why this is how the structure is made. Okay. Now, if you observe in this, you yourself can easily tell that whenever one end of the DNA is having 5 dash, then on the another strand of the DNA there is 3 dash end. Likewise, if here is 3 dash, then another end of the DNA is called as 5 dash. Please note the direction of all the oxygen. All oxygens, they are in the same direction. These oxygen atoms, they are in this direction, and these oxygen atoms, they are in this particular direction. That's why it is considered to be anti-parallel strands. Okay, this is how this DNA is. Now, if we talk about some of the dimensions, then between I'll write it with the help of another. If we talk about the distance between these two, then the distance between this base pair and this base pair, it is around 3.4 Armstrong unit. Okay. Likewise, this will also be 3.4 Armstrong unit. Likewise, this will also be 3.4 Armstrong unit. And if we talk about the distance between these two strands of the DNA, in that case, this particular distance, it is around 20 Armstrong unit. Okay, this is how the DNA it actually looks. But DNA is not a linear molecule. It is basically a twisted molecule. So how that particular twisting is done? Let's see. If we talk about twisting, in that case, the DNA it is twisted in an helical way. Now, what is mean by helical way? Helical way means, we can take for an example, like if you hold a paper, or maybe a stretch of the paper, and both the sides of that particular paper, they are considered to be the two strands of the DNA. If you just hold that paper and you just twist it anti-parallel, like one end of that paper you are twisting like this, and another end of the paper you are twisting like this. So this is how you are trying to twist this particular paper. In that case, it will be able to produce something like this structure. This kind of structure, it is actually called as helical structure. DNA is a helical molecule. Okay. So if we start, for example, if we start from here and then we try twisting, 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 and likewise, if we reach again back to this particular position, that means we almost covered 90 degree, 180 degree, 270 degree, and again 360 degree. This is how it is considered. So when a single twist of the DNA it is completed, like we started from here, we turned like this and we reached there, that is called as single twist. As there is this single twist is there, that means in one single twist there are 10 base pairs. In a single twist of the DNA, there are around 10 different base pairs. When 10 base pairs are there, when there is the distance between one base pair and the second base pair is 3.4 Armstrong unit, that is how the single twist of the DNA, it is around 34 Armstrong unit long. DNA is not only a single twist long DNA. So we are just taking it for an example. Like one single twist, it contains 10 different base pairs of the DNA of the nucleotides, and that's why it is 3.4 Armstrong unit long. DNA is a very long molecule, but this is one of the basic structure. So if we talk about the DNA, if we need to describe how the DNA is, in that case DNA can be described like this. That DNA is basically, if you talk about description of DNA, then DNA can be described as it is a double stranded polynucleotide with anti-parallel strands, okay, and at the same time it is helical structure, or you can call it as the helical molecule. These are, this can be considered to be the description of the DNA. Okay. Packaging of the DNA is something different. So today we will remain restricted up to only how the structure of the DNA is. Okay. This is called, this is how the DNA is. Thank you very much.